The overall goal of this chromatin immunoprecipitation procedure is to evaluate TGF-beta1 induced binding of the transcription factor SMAD2 to SMAD binding elements in the c-KIT promoter.
This method can answer key questions in the field of cellular biology, such as cytokine-induced gene transcription. The main advantage of this technique is that it has the potential to demonstrate direct binding of transcription factors and other transcription regulatory proteins to their DNA binding site in vivo.
Grow the cells to between 70 and 80% confluence on 10 centimeter tissue culture plates. Stimulate the cells per the experimental aims. Remove the tissue culture medium and add 5 milliliters of fixation solution. Then, incubate the cells for 10 minutes at room temperature on a shaking platform. Remove the fixation solution and wash the cells twice with 10 milliliters of ice-cold 1X PBS. Remove the 1X PBS and add 5 milliliters of glycine stop-fix solution. After incubating the cells for 5 minutes at room temperature on a shaking platform, remove the glycine stop-fix solution and wash the cells twice with 10 milliliters of ice-cold 1X PBS. After removing the PBS, add 2 milliliters of cell scraping solution and harvest the cells using a cell scraper. Then, transfer the harvested cells to a conical tube on ice. Centrifuge the samples for 10